Hello everyone, myself Manju Sharma from Suresh Kanvyar University, Jagatpur, Jaipur. The department is School of Agriculture. Today we are going to discuss about diseases of Crucifer, in which we will discuss about the major diseases of crucifers. So first disease is the club root of crucifers or finger and toy disease. The causal organism of the disease, that is Plasmodiophora brassicae, fairly several in hilly regions on cabbage, cauliflower and other crucifers. Once infected, field become unfit for cultivation of crucifers for several years. The symptoms are like infected plants have pale green to yellowish leaves initially, and later infected plants show wilting in the middle of hot, sunny days, recovering during the night. Stunning of above ground parts and reduction in size of heads. Characteristic symptoms become apparent in advanced stage of root infection as spindle-like spherical, knobby or club-shaped swellings. The swellings may be few or they coalesce and cover the entire root system. Basically, these are the symptoms of the disease. Finger and toy disease. So it will be looks like that. Survival and spread, like primary survival, soil borne resting spores which survive for longer period in soil like 10 years. Secondary autonomous zoospores also carried through irrigation water or by root contact. Favorable conditions, it occur at temperature range of 12 to 27 degrees centigrade, high soil moisture and neutral to acidic soils 5 to 7 pH. Their management, use of seedlings from disease-free field, plant cabbage and other susceptible crucifers, crops in well-drained field that have a pH slightly above neutral, usually about pH 7.2. Then add hydrate lime to soil to increase the pH of 7.2 like 6 weeks before planting, and it should be 2.5 ton per hectare. Avoid excess irrigation. Treat the soil of seed bed area with chloropicrin, methyl bromide or vapum 2 weeks before the sowing. Then the next disease is the downy mildew disease. The causal organism is Paranospora parasitica. The symptoms are like small purplish brown spot on under surface of leaves. Small, pale, yellow angular spot on upper surface of leaves with downy growth on the under surface. So the spot coalesces and the leaves revel and dries up prematurely. In cabbage, these spots expose the heads to soft rot. Cauliflower cuts look brownish at the top, then stems show dark brown and depressed lesions on streaks which later develop downy mildew, growth of fungus. So basically, these are the symptoms of the disease on the leaves. So there will be spots on the leaves and the color of the leaves will be changed, like it will be turned yellow from green color. Now survival and spread. Primary survival like oospores in infected plant debris or in soil. Secondary, wind-borne and rain-splashed sporangia. Then the favorable conditions. It occurs at a moderate temperature range of 12 to 27 degrees centigrade, then high soil moisture and neutral to acidic soils like 5 to 7 pH. Then their management, destruction of infected plant debris, avoidance of thick soil and excessive moist conditions, spray metal exile plus mancozeb like Radomil 0.2% or copper oxychloride 0.3% at 10 days interval. Then the powdery mildew disease. The causal organism of powdery mildew disease, that is Erisiphe polygony, infects mostly cabbage and cauliflower. Symptoms are like white powdery spots on the upper surface of leaves, stem, flower parts, etc. Finally, the mildew may cover the entire surface. Infected plants may show rotting and do not have normal growth. So basically, these are the symptoms. The leaf of the cabbage, which is like spores, will be also there, and it will be looks like the appearance insect bite. Survival and spread. Primary survival like dormant mycelium in infected plant debris. Secondary, wind-borne conidia. Favorable conditions. It occurs at a moderate temperature range of 12 to 27 degrees centigrade. Dry conditions with morning relative humidity of 80%. Then their management. Destruction of infected plant debris. Spray of Dinocap 0.2%. Alternaria leaf spot. The causal organism, that is Alternaria brassicola, Alternaria brassicae, and Alternaria raffini. Common on cabbage and cauliflower. Like the symptoms are symptoms usually on alder leaves. Spores are small, dark colored, they enlarge, soon become circular. Under humid condition, group of coniliophores will be formed in the spore. Then the next, spores develop concentric rings. Finally, the spores college, leading to lightening of leaves. Then short halls may be formed due to falling off infected spore tissue. The fungus is seed-borne and cause shriveling of seeds and poor germination. Now, linear spores also appear on petioles, stems and pods. So these are the symptoms like on the leaf this kind of spots will be there and the color of the leaves will be changed and slightly wilting will be also there. So that is the structure of the wilt, structure of the spore. then their survival and spread, primary survival, dormant mycelium or conidia on seed or in infected plant debris, and the secondary is wind-borne conidia. Their favorable conditions, relative humidity should be 80%, less vigorous plants and late in the season. Their management, hot water treatment at 50 degrees centigrade for 30 minutes. Next, seed treatment with Thiarum or Mancozeb. Then foliar spray with Mancozeb 0.25% or copper oxychloride 0.2% twice. The next disease is the white rust disease. The causal organism is the albigo candida. The symptoms are like white, shiny raised blisters, that is the pustules on the lower surfaces of leaves, stems and flowers. Then pustules coalesce to form irregular patches. The epidermis ruptures, exposing white spores, mass, which give the pustules a powdery appearance. Then the next is the distortion of the floral parts, including petals, pistils, and anthers, due to hypertrophy and hyperplasia. And the plants will be malformed beyond the recognition. So here, what is the meaning of hypertrophy and hyperplasia? So hypertrophy is when these cells in an organ or tissue increase in size. That means there will be a increasing in size, and there is the growth only in a particular organ or a tissue which will be increase in their size. So that is called hypertrophy. Hypertrophy, we can say that excess growth. Like if the lumberjack gets really tough, so that she can cut down twice as many tree. Another thing that is hyperplasia. Hyperplasia refers to the process where cell in an organ or tissue increase in their number. Hyperplasia, that means the growth will be in number, and hypertrophy, there is the growth will be not increasing the number, but it will be increasing the growth of the particular tissue or organ. Then next is the plants malform beyond the recognition. So basically these are the symptoms of the disease. These looks like a powder like appearance, and they will be appear basically at the lower surface of the leaves. It will be more visible. That is called white rust disease. Like white rust disease in will be the spots, the white color spot will be round in shape, or there will be a colony formation. Survival and spread. Primary survival, like the oospore in soil and sporangia from perennial weed host in the vicinity. Secondary, wind-borne and rain-splashed conidia or autonomous zoospores. Favorable condition like relative humidity 90%, high soil moisture, and frequent rains. The air management like crop rotation, destruction of fields, spray 0.8%, Bordeaux mixture, or copper oxychloride 0.3%. Now, the next is the diseases of mulberry. The first disease is the that is the powdery mildew disease. The causal organism is Phylacteria corylea. The symptoms are like the lesions appear as white to grayish powdery patches on the lower surface of the leaves. Gradually they turn yellowish brown to black patches, then powdery patches. Powdery patches cover entire leaf surface and make leaves unfit for feeding silkworms. The whitish powdery material seen on the leaves consist of the fungal hyphae conidia or conidiophores. These are the symptoms of the disease. Now, survival and spread. Primary survival like dormant mycelium or clastothecia in infected plant debris. Secondary wind-borne oval Areopsis type conidia. Their management remove and destroy infected plant debris. Spray of wettable sulfur 0.2% or Dynocap 0.1%. Then waiting period of 15 days after spray may be allowed before harvesting the leaves for feeding silkworms. Then the waiting period of 15 days after spray may be allowed before harvesting the leaves for feeding the silkworms. Now, the diseases of onion. The purple blotch disease, the causal organism that is Alternaria pori. Symptoms are like minute whitish dot on the leaves with irregular chlorotic areas on the tip portions, circular to oblong concentric velvety rings appear in the chlorotic area. Sometimes yellow halo is seen around the lesions. The causal organism is the Alternaria pori. Symptoms are minute, whitish dots on the leaves with irregular chlorotic areas on tip portion. Then the circular to oblong concentric velvety rings appear in the chlorotic area. Sometimes yellow halo is seen around the lesions. Then the spores will be colious and leaves die from tip backward. Disease leaf break at point of infection and hang down. Infection also seen an outer scale of bulb, seed stalk and neck. Then the severely infected crop that dries up, bulbs become dry and papery. Then these are the symptoms of the disease. Survival and spread. Primary survival like conidia in infected plant debris or seed bulb. Secondary, the wind-borne conidia. The favorable conditions are like warm, humid weather with rains or dew. And next is the thrips inspection. Now their management. Select healthy bulbs for planting and bulb treatment with mancozeb or thiarum 0.25% solution. Then the seed treatment with thiarum 3 gram per kg of seed, spray of mancozeb 0.2% or chlorother Then select healthy bulbs for planting and bulb treatment with mancozeb or TRM 0.25% solution. Then the seed treatment with TRM 3 gram per kg of seed. Then spray of mancozeb 0.2% or chlorothalonil 0.2% and then the seed treatment with TRM 3 gram per kg of seed, spray of mancozeb 0.2% or chlorothalonil 0.2% and then manage the thrips by insecticides. Next is the smudge disease. The causal organism is the Coletotrichum quercinens. Then the Isthmus disease, the pathogen is Coletotrichum quercinens. The symptoms are chiefly a disease of scales oval, red scale onions are then chiefly a disease of scales oval, red scales of onions are used to. Then the dumping of in seed bed under wet and warm conditions. Disease appear at all stages and also during the storage and transportation. Then there is the subcuticular dark green to almost black smudge appear on the bulb, neck or green leaves that the clinging to bulb after digging. Then the circular lesions with concentric rings of dark stromata and mycelium appear on leaves. Then the small, sunken and yellow lesions on inner scales. Then the disease appears at the all stages and also during storage and transportation. The subcuticular dark green to almost black smudge appear on the bulb, neck, or green leaves that are clinging to bulb after digging. Circular lesions with concentric rings of dark stromata and mycelium appear The subcuticular dark green to almost black smudge appear on the bulb, neck, or green leaves that are clinging to bulb after the digging. Circular lesions with concentric rings. The circular lesions with concentric rings of dark stromata and mycelium appear on leaves, then small, sunken, and yellow. Then there is the circular lesions with a concentric ring of dark stromata and mycelium appear on leaves. Then the small sunken and yellow lesions on inner scales, then pinkish mass of fungal growth on lesions uncle. Then the pinkish mass of fungal growth on lesions under the humid condition. So these are the symptoms of the disease. Basically, it will be looks like blackish in color. Then the survival and spread. Primary survival, stromata in infected plant debris or soil, secondary windborne cornelia, and the favorable condition are no, then that is the wet soil with a temperature of 26 degrees centigrade. Deer management. Resistant varieties like Nasik Red, Pusa Ratna, and Pusa Red. Protection of bulb from rains after harvest. Then production of bulb from rains after harvest. Bulb should be dried prematurely. Then the bulb should be dried properly before storage by and the protection of bulb from rain after harvest. Then the bulb should be dried properly before storage by hot air at 37 to 48 degrees centigrade. Spray of Ginep or Captain 0.2% before harvest of crop. Then next is the isthmus disease which caused by the Urocytest schifuli. The symptoms are fungus attacks cotyledons of young plant soon after their emergence, causing dark, elongated or eruptive spots. And older leaves, the lesions may extend from base to the tip and appear as blisters. Then the lesions develop into thickened areas of several in size. Lesions burst open, releasing masses of black smut spores. Then the severely affected plants killed within 3 to 4 weeks of emergence. Then surviving plants are stunned with stout, brittle, distorted leaves bearing lesions throughout their length. The Isthmus disease causal organism is Urocytes schifuli. The symptoms are like fungus attacks cotyledons of young plants soon after their emergence, causing dark, elongated, eruptive spots. Then, fungus attacks cotyledons of young plants soon after their emergence, causing dark, elongated, eruptive spots. Then, on older leaves, the lesions may extend from base to the tip and appear as blisters. Lesions develop into thickened areas of several in size. The lesions burst, open releasing masses of black smut spores. Severely affected plants killed within 3 to 4 weeks of emergence. Then, surviving plants are stunned with stout, brittle, distorted leaves bearing lesions throughout their length. Then, the numerous blisters appear on leaves and bulb scales of mature plants which rupture to expose masses of black powdery spores. So these are the symptoms of the disease. Basically, it will be black in color, survival and spread, like primary survival, spore balls in soil and seed bulbs. Then secondary, wind-blown soil and wind-blown wind-blown soil and seed bulbs. Then the secondary is the wind-blown soil and surface water having smut spores. Favorable conditions like optimum temperature of 10 to 1200 degrees, 10 to 20 degrees centigrade, and the plant is susceptible 4 to 3 weeks from showing. Then their management. The seed treatment with TRM 3 gram per kg seed before showing in nursery, crop rotation and use of disease free seedlings and seed bulbs. Then thorough application of sulfur dust 100 kg per hectare. Then the cultivar is hardy white, bunching is resistant, and spray with Captain 0.2% along with a sticker. Then their survival and spread. Primary survival, spore balls in soil and seed bulbs. Secondary, weed blown soil and surface water having smut spores. Favorable conditions like optimum temperature of 10 to 20 degrees centigrade, and the plant is susceptible for 2 to 3 weeks from showing. After that, their management, seed treatment with TRM 3 gram per kg seed before showing in nursery, crop rotation and use of disease free seedling and seed bulbs. Then Thorough application of sulfur dust 100 kg per hectare. Then the cultivar is hardified, bunching is resistant, and spray with Captain 0.2% along with a sticker. So this is all about the diseases of onion and crucifers.